Okay, I think we can start now. Hello, everyone, and welcome. It's my great pleasure to introduce our KITE Research Round speaker today, Dr. Stephen Scott, who will be talking for us on, who will be giving a talk for us, and the title is, Can Robotic Technology Bring Neurological Assessment into the 21st Century? Dr. Scott holds a GSK chair in neuroscience and is a professor in the Department of Biomedical and Molecular Sciences at Queen's University. He received his bachelor's and master's degree in systems engineering at the University of Waterloo and his PhD in physiology from Queen's University. His basic research program explores the neural, mechanical, and behavioral aspects of voluntary control, including studies on human and non-human primates. His clinical research explores the use of interactive robots and other advanced technologies for neurological assessment. He has published 170 journal articles and has given over 200 invited talks. He invented the Kinarm robot and is the co-founder and chief scientific officer of Kinarm, the company that commercializes these robotic platforms for basic and clinical research. Please join me in a virtual round of applause. During the talk and at the end, you can type in your questions in the Q&A box and on Zoom.
Great. I'll share my screen. Does that look good?
Yes.
Okay, well, I want to thank everyone for taking this time out of your day when it's so nice outside. Hopefully some of you are on your porches and outside and can enjoy yourselves in the sun. And it's good to finally give a talk here. This was scheduled back for April last year. And of course, that became a challenge for with COVID. And so we delayed, hoping we could do this in person again. But of course, this has been going on for a while, so I'm glad I have an opportunity to present, and I hope sometime in the future I can come down and actually visit Toronto Rehab and KITE. So first I like to do is a bit of a disclaimer, and that is I'm not a clinician. I'm a basic scientist with engineering and physiology as a background, so I don't have a daily experience working with patients. The other is that I'm co-founder and chief scientific officer of Kinarm that commercializes the robot. So I have a clear conflict of interest when I'm presenting, and you should think of that as you hear what I'm saying. But I hope those things actually help us in the challenges of translation. Hopefully the first, and it's commonly called the valleys of death between basic biomedical research, clinical research, and clinical practice. In the valley of death, one, hopefully the fact that I have a basic research background will help transfer some of the ideas, as I will present, to clinical research. And hopefully the company is a key thing to try to get through the second valley of death of translating ideas from clinical research into practice. Now, the short 2 slides of what my talk is about is really here. And this is the point that clinical assessment of body structure and function has made substantive advances when we think about blood-based biomarkers or imaging of brain structure. There's been huge changes of what is possible. But for brain function, it's still the same old of using visual and physical inspections by a clinician to score the patient's performance. And what I'm going to try to discuss this next hour is bring out the idea of maybe using interactive robots as a way to change neurological assessment. So I'll have 5 sections. They're not of equal length. Some are longer, some are shorter. I'll first talk about why I built the Kinarm robot in the first place, and that was to study motor function. I'll talk briefly about the potential of robots for clinical assessment. I'll then talk about how we've developed Kinarm standard tests and how we've applied it and used it to study stroke. At the back half, I'll have some small sections on areas we've gone to with the robots to look at beyond stroke, other neurological diseases and injuries. And finally, if we have time, I'll cover a bit of our future directions we're working on. So this all starts with my interest in understanding voluntary behavior, from the rather simple problem of reaching out for a cup of coffee, keep your caffeine levels high enough so you can listen to me as I talk here, But sports is one of my key areas. And of course, if you guys were probably a little sad right now because my picture here is of Carey Price, who is the last hope for us as Canadians to bring home the cup back to Canada after a drought of 28 years. But in sports like this, Carey Price gets paid a lot of money. He gets paid a lot so that if someone shoots a puck at him at 100 miles per hour, he quickly moves his glove, stops the puck, and does it in a blink of an eye of time. And that allows him to make millions of dollars and has the chance to win the Stanley Cup. Other sports also have more interaction. So football, there it's not just about individual person moving, it's how they all interact that matters the most. And these are the top ideas of how we move and interact in the world that sort of drove my interest when I started doing my graduate work and postdoctoral work. Now my postdoc, I actually was doing non-human primate work with a guy named John Kalaska at the University of Montreal. And our technology we used at the time was a pendulum. It had a potentiometer at the top, and the monkey had to move the little crosshair at the bottom of the pendulum to different targets located in the workspace, in this center outreaching task. Then we would record the activity of a neuron. So these little lines, each little vertical tick is an exponential. These are 5 repeat trials for movements to the left here, to the right, and away from the monkey. And you can see that this cell has a burst of activity, and we would relate the activity of neurons to the direction of movement using this device. Now, when I started to think about building my own research lab to study motor function and how the brain controls movement, I looked at what was available. And on one hand, there is devices like this that was the original device by Ed Evarts, where they did simple movements of a single joint. They would have loads. They could control the biomechanics and physics by changing the loads. It was easy to collect the data. The mechanical loads were easy to apply, and that matters for motor control because when you want to move, a lot of the problems about motor control is working with physics of objects and other things in the environment. But the behavior is really too simple. It's simply flexion and extension. And so you really can't get into a lot of interesting questions about how you move, redundancy, and multi-joint coordination. At the other end of the spectrum, there is devices like marker-based tracking, and you could have OptiTrack and you put markers on the arm and then you could measure how the arm moves. The data collection all of a sudden becomes a lot more complex to automate, which is required for doing neurophysiology. Applying mechanical loads is not easy. You still have a problem of how would you do that. The behavior is nice and rich, but it's really quite complex and almost overpowering for how much was required. And this is, we have to think back in 1995. So at that time, I was looking for what I call a 2-dimensional task, something that had a lot of the richness of 3-dimensional movements, but simpler. And so that's really what I was looking for as a compromise, something that would be moderately capable of applying loads, data collection, but have richness in behavior. And so I came up with this device called Kinarm, and nothing in science can be successful without an acronym. So Kinarm stands for Kinesiological Instrument for Normal and Altered Reaching Movements. And it's basically an exoskeleton. And you adjust the joints of the robot to line up with the joints of the monkey, the shoulder and the elbow. Arm troughs keep the arm onto the robot and allows the monkey to move in the horizontal plane. And so you can do combined shoulder and elbow motion to move your hand to different spatial goals. And with this technology, you can also use a virtual reality system where we projected targets down into the horizontal plane. So you actually reach 2 spatial goals in the workspace, not a computer-based task. And you can collect all this type of information. Where's the hands moving? What's the shoulder angle and elbow angle and velocities? And with fairly rudimentary equations of motion, we can compute shoulder torque and elbow torque. And soon after I built the robot for the monkeys and doing monkey neurophys, we started to flip over and want to do studies on humans. So we ended up building an exoskeleton system. And then when we turned that into having a company, we even built endpoint robots in many situations where you don't need as much complexity and richness of the information. You can get a lot out by just knowing where the hand's moving. So here's a little video to illustrate the system. This is the more newest system that we've developed in the last couple of years at the company. And in this system, you sit down, you can have the arm troughs You can see there, you adjust the robot to the geometry of the subject's arm, you align the robot, lock them in place. You then wheel them in so they can look through a virtual reality system, look down, and you can see the arms in this case. This is just calibration of the arm in the workspace, but then you can block it and then have targets projected down onto the workspace. And this is just the operator system. Showing a subject doing a task, the object hit task, which I will show later. And now they're just moving and interacting in the workspace. So with this type of platform, we've done a bunch of questions just asking how the motor system operates. How do we move and interact? And we've been able to think about taking real world problems like problems of reaching to doors, and that doors and door knobs and door bars are quite different. And if you have errors, you have errors and noise in our motor system, if you are reaching to a doorknob, you have to correct all those errors. If on the other hand, you're working a push bar, you actually have flexibility in the horizontal direction that you can exploit so that you can hit anywhere on that bar and the door will still open. We wanted to ask how fast could we make these types of corrections and whether they were different, whether you're reaching for a very small object or an object that has what we call redundancy in how you can solve the problem. So we converted that into a very simple dot and bar task. Here's just virtual reality system. The subjects actually only see the white dot representing their finger. Their arm is not there, you can see it. And then every so often we will perturb and mechanically apply loads and then look at the corrections. And what you'll see is these nice goal-directed corrections like that trial and this trial where the subject reaches the target. And here, if it's a bar, you see the subjects just allow their arms to be disturbed and go over to other points. So this is illustrating the point of redundancy. And this now lets us to go into try to understand how fast the motor system can do this. And if you look at the EMG, you actually find out the motor system solves this redundancy problem in 60 milliseconds. We've done other tasks here is like trying to avoid obstacles. Right now, the subject's reaching to a goal. The blue dots are obstacles. If you put your fingertip through there, the robot will push you back, so you have to avoid them. And again, we can look at these fast goal-directed corrections, how your voluntary motor system deals with complexities in the world, with putting small perturbations. Those were small. Here's a big perturbation that go around. All the last perturbations I'm going to show here are the same size mechanical perturbation, but you see people solve it different ways. Sometimes they go around, sometimes they go between. And it's instantaneous information they're using when they're moving to decide. And the 60 milliseconds of how fast the motor system can do is quite extraordinary. If you blink your eye right now, that takes about 200 milliseconds. But your motor system actually solves this in only 60 milliseconds. So incredible speed that we have for generating goal-directed corrections. And one of the things we can do with the non-human primates is we now can take those tasks and study the neural circuits that are involved in voluntary control. So here's a monkey that's been trained to do a simple postural perturbation task, and they make these nice goal-directed corrections back to the spatial goal. And then what we can do is record neurons. And so this is, in this case, a chamber we put over the brain. We can drop microelectrodes down and record from neurons in the brain, whether it's in primary motor cortex, premotor cortex, parietal area 5. And this is just illustrating two neurons that responded very briskly to a mechanical disturbance. So the disturbance was applied on this 0 milliseconds. And you'll see the discharge rate increase in both primary motor cortex and parietal area 5 in about 20 milliseconds. You'll see activity in these cortical areas. So cortical or sensory information can get up in 20 milliseconds. And actually it gets down in 60 total milliseconds in giving you these goal-directed corrections. Last little study from looking at fundamentals about brain circuits for voluntary control is actually one that's kind of related to thinking about stroke. In this case, it's a situation where we apply cooling probes and we place them right on the surface of the cortex. It actually allows us to do temporary lesions by passing methanol through the tube. We reduce the temperature of, say, premotor cortex, this tiny area right here. We can stop all neural activity and then look at how the monkey behaves and how does that impact their behavior. So we put cooling probes either in premotor cortex or in parietal area 5. And then we can look at the impact. So before the cooling, this is the monkey's behavior. This is work by Tomohiko Takai, who was a postdoc in my lab, who's now back in a faculty at Kyoto University. And during cooling's over at the rate. And what you'll see is there's a little bit more difficulty in performing these tasks while this area of cortex, in this case, premotor cortex, is cooled. If we go to the raw data, this is the corrective movement that the monkey makes. This is before cooling, this is during cooling, and this is after cooling. So we turn it off within 5 minutes, the brain area is back to being active. And you can see these are much better corrective responses. This is the hand motion. You can see that the time back starts to become more variable. Endpoint errors start to go up. They can't keep their hand at the target as well. And that's just illustrating here how the endpoint error goes up during cooling as compared to if it's a sham where we did everything but actually turned on the methanol. And you can see the performance is much worse, whether it's the speed or the accuracy. So here's a way that we can make temporary lesions. And one of the ways we want to go forward is to actually look at how does that impact neural processing in different brain regions. Now, the next thing I want to shift to is to look at robots for clinical assessment. And as all of you, I'm sure, know, patient assessment drives patient care. Does Sidney Crosby have a concussion? Is this drug going to be effective to improve or ameliorate the effects of a disease or injury? And if you're going to create a rehab program, is strength the biggest problem? And so the idea for clinical assessment is to kind of use the robot the way I use it for my basic research, or kind of a reverse way. I have an area like premotor cortex. I have a number of, I developed behavioral tasks. And from that, I I have tasks, and from that I identify brain function. Maybe I'll stop there and respond. Will you be able to add EEG to the system? Yes, some labs actually do use EEG, so that is certainly possible. I haven't done it myself. And the monkey, male or female, most of the monkeys have been males in the past. We are now starting to flip to using females. And I do not expect any differences between them in their circuits. So those are just a couple questions that popped up. So what do we do for clinical practice? What's wrong with his brain? Well, trying to use the same way that we use it for basic research, we can build a battery of tasks to study different brain processes, and then have subjects perform the task, look at the pattern of impairments across the different tasks that are targeting different brain circuits, and from that be able to identify, what are the dysfunctions, and even maybe what are the brain circuits or pathways or regions that are impaired. Thinking about robotic assessment, and one of the big things is to think about going beyond just studying folks of spinal cord and primary motor cortex. A clearly important thing for assessment is to look at all parts of the motor system, particularly related to something like stroke. And so there's a lot of effort to try to cover all the mechanical degrees of freedom of the joints. And that's absolutely important. But robots aren't going to be that useful for trying to do that type of analysis. Rather, they're useful for all the other circuits that are involved in perception, cognition, and motor actions, voluntary planning, online control, perception of our body, and execution and decision-making. Those are the areas that really, I think, the robots are going to be able to exploit and give a lot more information. Why start with expensive robots? I hear this all the time. Is an assessment of the future, we're going to use phones, Fitbits, and these are much cheaper devices. And our strategy really is quite simple. We don't think there are any gold standards. Right now we have bronze standards to assess brain function. And the challenge with using inexpensive devices is cheap constrains potential. So we need time to discover with any technology and expensive technologies, what is the best things, what can we assess? And when we do find those things out, then the next stage can be what tasks are required and how can we make it cheaper? So it's this phase of trying to develop the gold standards, the best way of assessing the brain that we're in at this moment, or at least that we're focusing on. Another challenge in trying to do this work is this problem of the valleys of death. A common way that people like myself in basic research, we have this task like making goal-directed corrections to avoid obstacles. And the natural question is, oh, some patient groups, say strokes, they're going to have problems with this. And so they go and take the task, make subjects perform the same tasks. It's going to take 30 minutes for you to collect all the data. Best you're going to get is group effects because you're going to just do an age match control. And that works to try to translate from basic biomedical research to clinical research, but that won't translate to clinical practice. The requirements are completely different. Patient-specific impairments, it's not about group effects. You want to know what this patient has and what their impairments are. The instructions have to be really simple. If they're too complex, it doesn't work. Time is your enemy, so these tasks have to be short. And there's always the cost and value benefit that has to be thought about. And so we've come up with a standard way of looking at these where we think about building a task, and it's always thinking about a brain circuit that we're interested to understand. We develop a behavior, we go back and forth trying to hone in on a simple task, simple instructions that takes little time. Once we finish that, we have some data. We then take apart the spatial and temporal features, in this case of the hand movement going to the target, and this is the hand speed. Pull out a number of spatial temporal variables, collect hundreds of healthy control subjects, because a core to our approach is to not just make it ordinal, but to have it based on how do you perform as compared to what a healthy individual behaves. Take all the healthy controls, identify normative models to say what does healthy performance look like, where you correct for things like age, sex, handedness, and then you can push through the patient data and give them a score relative to what healthy subjects should perform like. So with that as some of the principles of how we go about the assessment, I'll switch to talking about the tasks we've come up with, and in particular, their application to stroke, since stroke was really the key area we've worked on. And so this is the battery of tasks we've come up with. We can think of them as cognitive, motor, or sensory as far as what they're trying to get at. I like to think of it as a triangle, because really every task has some sensory, motor, and cognitive. It's just that some of them emphasize one aspect more than another. And so we've got about 12 tasks at this point, some more motor, some more sensory, and more cognitive. And a lot of this work is done with Dr. Sean Duclos, a physiatrist in Calgary. He did a clinical residence at Queen's and was a postdoc in the lab, and we've continued to work very closely together on this project of developing behavioral tasks and using robots for assessment. So we really have a convenient sample. We get as many subjects as we can through the robot. We'll pick ischemic or hemorrhagic, and the subacute early group is usually somewhere between 7 and 28 days. And these patients can be collected at one of 2 sites at Kingston or in Calgary, but the majority of data is coming from Foothills Medical Center, where they have a very large cohort of stroke subjects going through that hospital system. So the motor tasks, this is just some example videos of some motor tasks. One is this bimanual task where you have to try to move a ball around the workspace and coordinate your 2 hands together. So this is a ball on bar, we call it. These 2 on the right is a visual guided reaching, and this rapid motor skills, we call it an object hit task. And I'll show a bit of data on those on the next few slides. So here is just to illustrate a single parameter from this reaching task where you have to reach to a spatial target. What we look at is the initial direction error. How straight did you move towards the goal? We take all of the healthy control data, normalize it, and that puts all of healthy subjects in a standard normal distribution, which is Z units. Basically all healthy subjects, 95% of healthy subjects will be between -1.96 and +196. So this gray area down here is what healthy performance is for this score. And now we are showing the initial direction error for the contralateral lesional arm. So that's the affected arm, and the ipsilateral or the unaffected arm. And these are the scores. So these subjects actually are within the healthy normal range. You see subjects have scores up to about 15 Z units. These are severely impaired. They are hardly moving at all. Likely aren't hitting any of the spatial goals, move very slowly and very jerky movements. And then you see this nice resolution that you can get because it's an analog score. What's also interesting is you can see that there's actually quite a few subjects that have impairments of both arms, so that it's not just the contralesional arm that's impaired. You can score and quantify many having impairments in both arms. The value of some of having these nice coarse or non-coarse scales is illustrated here. This is a subject that had an assessment 4 weeks post-stroke and 5 months post-stroke. These are the Shidokuk Master scores. This is a 7 and a 7, so that is perfect performance on the Shidokuk Master of the left arm. The right arm is 3 and 2, so that's fairly moderate to probably severe impairments. What you can see is after 5 months, they didn't change at all using the traditional scores. But if you look at the data, you'll see that they actually are getting better. Here you can see for movements away, they clearly are doing a much better job at moving towards that target. Lower here is also doing much better, and there's less variability. And so when you take the task score, you can see that they actually have improved from a score of 5.6 to a score of 4.5. This was a subject, very early prototype task we did many, many years ago, where we actually had 16 spatial targets. And this was a subject that came in, he's a chronic stroke when we were prototyping the system, and was quite upset because they took his driver's license away and he didn't understand why he should have that done. And if you look at this reaching data, you should be able to guess why they took his driver's license away. He has clear neglect from movements over to the left. And so in other words, he can drive and he will see a pedestrian coming from the right side, but coming from the left, he's clearly going to have difficulties. And so there's a really good reason why he doesn't have a driver's license, and a simple reaching task can tell you that. How do you separate learning effects when repeating the assessments? It's a great question. We've actually done tasks or repeat tests. So we've put the same people through the system twice, and we've now quantified the learning effect. All of the tasks have minimal to no learning effects. The one task that's different is something called the reverse reaching that has a small effect. So that's an important thing to try to get how much the learning effect is. And so that's actually a study that actually someone that's on here, Leif Samanis, put that study together to look at that effects of learning as well as confidence intervals and significant change. Good question. So we wanted to go in with this spatial neglect, a more recent study. This is work by Kane Park in my lab. I should add a picture on here. Oops, and I needed to spell better. Quantify reaction time across spatial directions. And so Kane's been working on a way to score spatial neglect. And we came up with 2 different metrics. One is the asymmetry in reaction time. So we looked at the reaction time in different spatial directions when subjects reach. And this is showing that if you have a reaction time that's much longer for, say, for movements to the left than to the right, you get an asymmetry score of like 0.5. That's what this 0.5 is, this distribution of reaction times. This is 0.25. So we get an asymmetry score. And then what we do is we look at a general score for the fastest reaction. And that is that we look at the opposite side to the direction that's slowest. And we look at how fast are you in the good direction. And this is just showing you a general score going from 250, 500 to say 1,000 milliseconds. So this is scorings for a healthy control. What you will see is the reaction times are very similar. The lengths of these arrows are each one's representing the reaction time in different directions. And so there's uniform reaction times in all spatial directions. And so there's no real asymmetry. And the reaction time in this case is going to be a few hundred milliseconds. Here we see an example of a left affected stroke. So the left arm has, I'm not sure it's supposed to, well, basically it's to show that both the left and right arms are impaired. This is the reaction time, and you can see the slowest reaction time was for movements down and to the left. Use of the individual trials here. And what happens is that on some trials, the subject never actually reached. And so they just got a score of 3 seconds because 3 seconds the light was on and they didn't respond. So you see this impairment in that direction. For the right arm, which is the unaffected arm, you can see, you still see that spatial neglect, although see it rotates a little bit. So now the maximal direction that you have slowed reaction times is straight to the left. So there's a bit of a rotation here. Here is now the reaction time impairments for that asymmetry score. And so healthy subjects, this is a polar plot. The direction is the direction where there is the slowest reaction times. And then normal range is the dashed line. So any number inside that range is within the normal healthy range for a symmetry score. All of these subjects out here are asymmetric as far as the reaction time is slower, and they are slower generally over to the left and towards themselves. For their unaffected arms, you see it's actually to the left, but actually a left and upwards to some degree. So there's this nice egocentric rotation that occurs between the left and right arms. These were only the left affected subjects. Right affected, of course, there's less people that tend to get spatial neglect, but there's still a substantive proportion. We can get about 20% that are identified as having visual spatial impairments. This plot simply compares the asymmetry score versus the fastest. These people down here actually have no asymmetry. Their reaction times are slow in all directions, and they have similar reaction times. So that's a general spatial neglect. And this is individuals that have neglect or neglect that's in a very specific direction and not general. And these actually have a mix of the two. So these two scores give you a nice way to quantify the two different features, if you want, of spatial neglect. Does the exoskeleton prevent compensatory movements? For example, moving the trunk instead of the arm? Yes. So one of the things with the exoskeleton, we can have a harness. It actually means you do really need to use the shoulder and elbow for performing the tasks. The other small point to point out here is that the size of the circles are for different bit scores. Bits below 130 are generally defined as having spatial neglect, whereas ones higher are not. And what you can see is some of these circles are actually not the large ones, which means these people pass the bit, but actually in ours show they have substantive spatial impairments in responding to some visual goals. Oops, I need to get. This is the object hitting task. So another fairly simple, and again, this is one of the examples of a task that's very easy to perform. You simply hit objects as they move towards you. You have paddles representing where your hands are in space, and then you simply have to hit them away. And the task starts nice and easy, but with time, things get faster and faster. And now you'll see that this subject that's healthy uses the full range. Here's a subject with stroke and doing a rational choice where you're seeing they're using their impaired arm, they're just using a small part of the workspace, and their better arm, they'll try to use as much as possible. So we can quantify that asymmetry. And then here's a subject with spatial neglect, as you'll see that they really don't even pay attention to the left side and get completely focused on the right, even though this is their good arm being pinned over to the right side. We also have perceptual, sensory and perception for action tasks. We have a limb matching task. I'll show a bit of data. We have a postural perturbation study. And actually with George Moshizuki, we did this elbow stretch test with the robot that's sitting at Toronto Rehab Institute. So we looked at that to try to measure spasticity of the elbow. This is the arm matching task. The robot moves one arm and the subject has to do a mirror match to assess position sense. There is a bibs and the subjects can't see their arms at all. And so we are just looking at their proprioceptive function. And then they simply look at where the targets are. This is the black dots are where the subject should have put their hand. The blue dot is the mean position they did for each of those trials. And the circle is the trial by trial variability. So you can see with this stroke subject that the ellipses are large, reflecting that from trial to trial, they go put their hands in very different locations. And you can see a shifting of the whole workspace away from themselves. What's interesting with this task is that you can see various types of impairments. It's not just that they're bad at the task. There's different characteristics. This is an MCA stroke, large circles. That just means they're guessing and have very poor information at all. Here you see subjects that actually have very small ellipses. They have very good understanding of where they want to put their hand and they pick the same location each time. But the workspace is shifted either in this case towards themselves or to the right, or in this case, a situation where the whole workspace is contracted. But again, they tend to have fairly small ellipses saying they pick the same spot each time. If you go within 2 weeks of post-stroke, 70% of subjects are impaired. And some work that Sean's been leading has been using voxel-based imaging techniques to identify specific areas that are important for this task. And parietal cortex and insula both get flagged as if you have lesions there, you have a high probability of having impairments in these tasks. Here's a control subject that we had go in the robot. This is their matching task. This is where they put their hand. This is where they put the spatial, or where their hand should have been. We were a little shocked by the performance, and so we talked to the physician. They did a CT scan and found a left cerebellar infarct and a dural-based tumor. This is a postural perturbation task. So this is a very simple one, sort of from my basic research at looking goal-directed corrections where the subject has to quickly come back. This is healthy performance. This is a subject with impairments, and it's simply showing how you can, again, score sensory use for motor actions. What we found interesting was that both arms often are impaired. Both the unaffected and affected equally can be impaired, not just a small impairment with the unaffected arm. They can be both impaired equally. We also go into cognitive and cognitive motor type tasks. So we have an object hit and avoid, where you now have to hit objects, but you have to identify the correct objects and have inhibitory control to avoid the ones that you're not supposed to hit. Or we can put tasks, pen and paper tasks like trial making onto the system as well. So this is an example of this reverse reaching task, one that gets at cognitive motor, and it's a powerful task. This has been introduced by Lauren Sergio, came up with this task from York University, and we've put it on the system. Healthy controls quickly learn to move the cursor in the opposite direction to their hand. Again, the subjects can't see the hand in this that's covered. They just see the cursor. But you can get these subjects with stroke and other diseases have enormous challenges understanding what they are supposed to do in trying to guide the cursor to the target. This is that subject that was shown on the previous slide. This is their performance during reaching. They actually have very mild symptoms. Their MoCA is 26, their Shidokri master, they have some impairment of the hand, but not the arm. Reaching movements were really quite good. They were slightly slow moving, so their movement time and speed were lower than normal. As soon as they do the reverse reaching, you can see this data here. All of a sudden, they completely are incapable of having their cognitive systems control their automatic motor system. It's just completely fallen apart. And you can see, in this case, very severe on the affected side and some impairments in the unaffected, but it's actually asymmetric control. So we've been looking at this. This is work Catherine Lowry in my lab has been starting to put together. This is the reverse visual guided reaching task. This is the affected arm. This is the less affected arm. And this is the scores. The healthy range is this bottom box here. This is those that are impaired just with the affected arm. And you can see there's a very large number of subjects, almost all stroke subjects fail this task. And they can fail it either with their affected arm or equally with both arms. We've also been looking across time. You can see the less affected arm is getting much better. Those that were very impaired improve. What's quite surprising is that with the affected arm, we're not seeing that improvement. And this is a group of subjects going out 6 months. So this impairment is remaining. This is looking at reaching versus reverse reaching. And again, you can separate that these people have motor skills for reaching on top of, then they, of course, not surprisingly have troubles with the reverse. These subjects here, on the other hand, have fairly good motor skills. They're only mildly impaired, but now very impaired in the cognitive component of this RVGR. And you can see that for the less affected, even more that it's commonly the RVGR task where the impairment is, and less impairments with just the standard reaching task. So those are the tasks. And one of the things with all these tasks is not just the value of each task, it's actually the value of using them all together so that we can go across sensory, motor, and cognitive. And we basically get to see a whole phenotyping. What's their pattern of impairments across all of the circuits and all of these different motor functions? So this is now looking at the scores. Task score is simply an aggregate of all of the individual task scores, parameters put together. Anything over 1.96 is impaired. And you can see this subject has a lot of impairments, some worse in some tasks than others. And now you can take that across and look for what kind of patterns exist in stroke subjects. Here's looking at two subjects, and you can see they have very similar patterns of impairments with severe impairments in reaching that carries over to the reverse reaching. But you see less problems with executive function, less problems with proprioception. And then we have two patterns that are quite different. You see this one where there's really more as you get towards the cognitive tasks, object hit and avoid, trails, spatial spatial working, that's where the working memory, that's the impairments. And this one that also has very different impairments, very much object hit and avoid and reaching. And when we look at all the subjects, we kind of find about half the subjects fit this standard pattern. And then the other half all are sort of unique patterns that are quite different from each other. So that's an ongoing sort of project. And with all this, it's the question with all these different tasks as we start to layer out, look at the different types of impairments on their spatial and temporal characteristics. When we're looking, traditionally, you're looking at strength, reflexes, coordination, spatial neglect will often get assessed. But there's a lot of other things going on with these subjects that have problems in executive function, inhibitory control, attention, motor planning. And so it's those things that the robot hopefully provides value. Oops, do I have a question? Something came up and then it disappeared. Maybe I'll keep going and we'll try to get to that. So I'll try to cover a bit of going beyond stroke, and if we have time, a bit on future directions. So one of the things is that the design of these tasks was really to build tasks to look at brain circuits. And so they're potentially applicable not just to stroke, but to lots of other disease. And so we put a lot of time, and other groups that have these Kinarm robots around the world start to look at the potential use of these types of behavioral tasks in areas like concussion, development, Parkinson's disease. So the hope is to see this as a very general tool and have it look what its value is for each different condition. I'll just give a couple examples. This is a work from Al Jin's lab. This is the difficulty of identifying a TIA. This is if you want a transient stroke, 60% of suspected TIAs are non-TIAs, and 60% of those are migraine. And so this issue of how do you separate different groups, this is again Al Jin and actually Leaf, who I think is on, and now a postdoc at Toronto Rehab Institute, worked on this problem. So we collected a large number of TIA patients. These are subjects that only had symptoms for 24 hours. They had no lesions from imaging, and they were assessed 4 to 14 days post-event, so well past the time they had any symptoms. These are their performance across motor, cognitive, and sensory tasks. This subject looks fine and it was well within the normal range. But you can find a number of different subjects. In this case, this subject had several impairments in the reverse reaching, trail making, and even some of the motor skills, quite severe for the reverse reaching. And then also this subject, you can see having impairments. And then what we can do is go across all the participants. So these are all the participants on the x-axis. These are all the different behavioral tasks on the y, and they're color-coded for whether they passed or failed. So anything with the red is that they failed the task. These are the TIA patients, and these are the migraines. And the first thing you see different between the TIA and migraine is clearly TIA have a lot more people find impairments, and they find impairments that can also be observed in the motor, this group here, far less in the migraine. Although there is an interesting large number of reverse reaching that migraine subjects, that means they had a migraine. Again, this would be 4 to 14 days. They may have had migraines in the past regularly, but they actually tend to fail much higher than by random chance. Only 5% of subjects should randomly fall into above 1.96. So more than that. Another group led by Ron Levy, and the student is Pauline Grappillin, and that's on Parkinson's disease. And so they've been working on using it to look at the benefits and the drawbacks of therapeutics for Parkinson's disease. This is looking at the reverse visual guided reaching task. This is direction errors, and this is reaction time. So for the reaction time, what you can see is when you go from baseline to drug, there's a huge drop, or there's always a drop in reaction time. There's a large one when these people, they're very slow. But even for those that had pretty good reaction times, they got better with drugs and DBS or deep brain stimulation. But the downside is direction errors, that instead of moving the cursor in the opposite or moving the hand in the opposite direction to the target, they actually would always try to move the hand to the target. So they would get a direction error. And you can see that the number of direction errors goes up as you add drug, DBS, and both. And you can also see that in the bottom diagram. So you have improved motor function, but you have a problem with inhibitory control starts to be a problem. Object hit and avoid. This is again this task where you have to hit some objects and avoid others. Number of correct object hits systematically goes up as you do the therapeutic interventions, but the number of distractors, things you're not supposed to hit, also goes up. So two different ways of quantifying that, the benefits and the drawbacks of drugs and DBS for Parkinson's disease. Last group I thought I'd quickly show, although I'm going to run out of time, is just to illustrate another group that's not neurological, and those are patients with kidney disease that are just about to get dialysis. This is a number of different tasks on the bottom here, and these are the scores for each individual, and you can see that a large number of subjects with about to get kidney dialysis actually are failing their tasks. That is because the urea that builds up in the blood actually has a detrimental impact on the brain function. And these are just some example profiles for individual subjects from this study. So last, I'll try to just do a couple slides on future directions, but keep it simple. We're going a number of different ways right now in trying to move things along. One is that we've got very large data sets. And so the value of that is that we can start to look at things like autoencoder-based classifiers. So instead of just scoring using temporal and spatial parameters we decide upon, we can take the raw data. We can use that raw data from, in this case, a large cohorts of controls or strokes, thousands of trials, and we can develop autoencoders to either do better classifiers to say, are you stroke or not? And then quantify and classify that. The other area we're doing with these are support or self-organizing maps, where we can now try to look at where people fall within some distribution. And that is all just organized by their performance and skills. And this is these self-organizing maps. This is some data we're just working on. It's still pretty early days, but at least some interesting ideas. This is Shidwoka Master stroke assessment scores from 1, which is the most severe, 2, 3, 4, 5, 6, 7 is a perfect score. In theory, you're supposed to be the same as healthy controls. Healthy controls would get 7. And this is the distribution of all the subjects from each of those. The classifier and the self-organizing map did not know what the Shidwoka Master score was. It was only generated by the reaching data. You can see that all the subjects that have a Shidwoka Master score 1 land on this part and are positioned on this part of the map, which is diametrically opposite of the healthy controls in the opposite corner. Subjects that are 2, so slightly less impaired, you can see are shifting over here. 3 now actually shows 2 different clusters starting to emerge, with the question of whether, even though they got a score of 3, are these 2 very different phenotypes or endotypes that emerge at this point. 4s and then 5s, you start to see a far more broader dispersion of patients, reflecting the point that they have, even though they all get a 5 with the Shidwoka Master, they have a range of performance capabilities all within that one cohort. And then we see 6 and then 7 as it starts to come towards a healthy control. Another thing we're working on is increasing the number of behavioral tasks. So we are continuing to increase the repertoire. We have a fast interception task where we can do visual, mechanical, or even in cognitive rules change. Oops. We have other reciprocal movements, tracking tasks, more sensory tasks, and we will be continuing to build up the cognitive tasks. And the idea is to have a very large repertoire that are available, fully automated. You can press a button to run the task, press a button, and you get the report. Now, all these tasks create problems, and that is time. And so one of the things we have to start, and we have been starting to work on, is how do we save time in the assessment? One is to create hierarchies of behavioral tasks, where we do a few tasks, build expert systems that then pick which task you should do after that and ignore the rest because there's no reason to assess them. And from that, build an expert system that then tells you exactly what the problems are and potentially what's the problem with the circuits. Also, the idea of, can we stop tasks early because we already have enough information, we don't need to continue and we can move on to the next task, as well as trying to make disease-specific tasks where if you have Parkinson's disease, we really know we should really focus on these specific capabilities. We're increasing technologies, not just the robots, which we use a lot, of course. We've got eye tracking built in that can be integrated, so you can look at eye movements on their own, but also while you're doing movements of the arms. We're also developing markerless motion capture systems with a local company in Kingston called Thea to try to get all the things that I can't get with the robot for whole body movements, postural control and balance, Markerless motion capture, I think, is a great opportunity. And of course, simpler tech for many applications. And when we're thinking about these, from simple tech to complex, we really see the robots as trying to create that gold standard, which is highly comprehensive assessment capabilities, high resolution, and great precision, but they do cost a lot. There's a lot more simple tech, and there's a lot of interest in that. And they should work together. One is the gold standards of the robots help identify potential biomarkers in these simple technologies. And in thinking about a clinical outlay of these simple techs provide early warning devices to provide effectively a triage for individuals that may have something happening that justify coming in and getting a more detailed examination. Will robots ever be used in clinical practice? Well, I don't know. I think at this point it's working well for a clinical research tool to try to understand what's impaired. The moving to clinical practice really comes down to a cost versus value. And it's not like cheap is the way it always has to go. This large donut over here, the MRI machine, is incredibly important and incredibly expensive, but it's really useful for guiding practice. And so if robots or other types of technology are ever gonna have impact, they really have to demonstrate the value to the healthcare system. With that, I want to thank the many people in the lab that make this possible. I have a large research staff that help run a lot of the systems in the hospitals and at Queen's. A large number of subjects involved in these studies. Leaf is in here somewhere. He's a postdoc. He did a lot of the studies with the robot. And a number of Kane's work was shown. Faranack was doing the autoencoder work. And with that, I'm take questions and looking forward to hopefully working more with Mark Bailey and Sandhya Gopal at Toronto Rehab in the near future. Thank you for your time.
Wonderful. Thank you so much for a great and fascinating talk. Thank you on behalf of our institute and everyone in the audience. I think at this point, everyone in the audience, you can either raise your hand. If you have questions, you can either raise your hand or you can type in your questions in the Q&A box. I think you answered many of the questions during the talk, but I think there are still a couple in the Q&A box that.
Yeah, okay, let's go. Does the exoskeleton prevent? I did that one, didn't I?
You answered. Yeah, I think, yeah, that's one of the ones that you answered.
Is head position fixed? Can they rotate their head, neck? And if they can, will this impact how neglect is measured? Right now, they have full freedom to move their head around. So they just move naturally how they want. People just keep it in one position to look down at the screen because they're usually working in this part of the workspace. So there's usually very little head movement. But it is a very interesting question on whether you had rotation of the head and neck and how that would affect the neglect. One of the interesting things from that study I found was the fact that the neglect actually rotated when you went from reaching in this part of the workspace to here. So that's really interesting to know. Is it body centric? It could be head centric, but it looks like it is, at least a large number of those subjects, that's what it looked like. So that is something to look at. We'll probably get it down that they will maybe not be able to move their heads, but again, most people just keep them fairly fixed to keep looking down.
Very interesting. Before you answer the next question, can I jump in and ask a quick question?
Sure.
Other than the robots, what other data do you record other than the movements of the robots? Do you video record the participants, for example, or any sensors on their body? Or
at this point, I have tried to develop all the platform for these Kinarm standard tests with nothing else. And the reason for it was to make it so it was fairly robust and able to do that. The system has analog channels and there's lots of TMS capabilities. So particularly when you're starting to use those other technologies, you can custom make your own task, integrate that with other platforms, bring in and link up EEG, TMS, postural control. When we have a standing version, we have a ground reaction force that all that data gets collected in while you're standing on the force platforms. So this is where the clinical side, we've tried to make it locked down and standardize. The basic research, or even a lot of clinical researchers, they can add EMG, they can add any and effectively any other devices with the system, and then they can analyze the data. The moment that happens, then it's much harder and people have to analyze their own data. The Kinarm standard test, the whole point is that you press a button, it's fully automated, it's fully normalized to everything's done so that you just have to look at the patient's performance in these scoring systems. So they're kind of two different ways of doing it. For
research purposes, if someone decides to collect also different modalities, do you provide functionality to synchronize with Kinarm data, or is it like a signal to?
If you make your own task, so if you want to make a task that, say, let's take TMS, so that you want the TMS to occur just as the subjects, as you turn the light on or in the middle of the reach, you can do that. It's all programmable. So we've got a full programming language. It's all in Simulink and Stateflow. So, and even embedded MATLAB code can be used. And so you can make your own tasks. And that's what, we do for a lot of our basic research. And so you can do that, it saves it all. Sometimes you have to sync it. It depends on what you're working with. A lot of my microelectrode arrays are a challenge because I have hundreds of electrodes, and that's got to go somewhere else. So now we have to sync with them. But all of that's generally possible. It's all about what your expertise is and what your science question is really what it is.
Very cool.
What else do we? Q and answered. Oh, there it is. What's the next question? What percentage of prisoners with stroke will perform within the normative range and control within the abnormal sensitivity specificity test? So we have gone for the abnormal. So the healthy controls, we use a standard line of 5, the 95th percentile, if you want. So we use that as a normative. That's the 1.96 in our task scores. And so 5% fall out of the normal range. There is also the fact that we do get some subjects that are outliers, of course, because some subjects might have underlying problems. And in fact, a task like reverse reaching is so sensitive. We know from Lauren Sergio's work that people pre-Alzheimer's and pre-mild cognitive impairment, just having children of people with Alzheimer's that have been assessed actually start to have impairments. So that's an interesting, challenging problem of some of these tasks are quite sensitive. So then you get into what's normal. For the strokes, it depends on the task, how many fall within the normal range. Some subjects do, but we also tend to put subjects in that have strokes that have mild or no impairments on traditional scores. So if a subject has impairments in the leg and the stroke really only impacted the leg, we often will still assess them to see if we see anything residual in the arms that can be observed. So even though they get Shidoko master scores of a 7 that says there's no impairments, we find many of those are impaired, and our new autoencoders find we can pick off about 60% of those. I think it's about 60% will be identified as impaired, even though they don't have any impairments with the traditional clinical tool. So it really varies.
Very cool. There was also another question and answer from Kinarm that responded that yes, non-supported peripheral data, EMG, TMS, EEG, etc., can be synchronized during Kinarm data collection.
Yeah. Do you think performance on the test is affected by psychological factors, computer skills, interest in performance is about links between tests and daily life? Yes. One of the things I wanted to negate is the impact of computer and gaming experience, because you watch someone that does not use a mouse. My mother has never learned to use a mouse on a computer properly. It's quite painful to watch. And that's because it's a skill of moving your hand down here to an object moving on your screen. So one of the reasons for that virtual reality system is you simply reach out to things that are sitting in the world. It's virtual, but they are spatially where you have to move your hand. So someone like my father, who is 88 and has gone through our robotic system, he has never used a computer and quite proud he will never use a computer in his life. And he performed well within the normal range. He had no problems. So that kind of gaming skill does not seem to be an impact. Interest in performance always is a challenge. Malingering, this is actually when we go to do concussed patients. We've worked with Brian Benson and Sean Duclos to do football players and other elite athletes in Calgary. And the worry is always that they're malingering. They don't want to perform well in the baseline test. So if they come back and have a concussion, that they have a less chance of failing. So that is always a major challenge depending on the group you're working with. But you got to keep it interesting, and you have to be careful that it doesn't get too challenging. So that's part of the secret sauce of how do you make a task that will keep them engaged enough that they will do that. We've had some challenges in our spatial memory, working memory tasks, because by definition, they can get hard and that can get frustrating. So it's always trying to keep that enthusiasm up. Can you comment on the possibility that the test results from the upper arm could translate to understand the impact of the function of lower limbs for walking, balance, mobility? Yeah, thanks. Yeah, one of the things I really was interested in, and I'd love to, so much time, no time and so many good questions. I really liked when I perturb one hand or when I have a stroke, that both hands were equally impaired. And that is, some of these rapid corrective stuff is probably involved in premotor cortex and other areas, and it's very bilateral. And so that's maybe why you get impairments on both sides. So that then also made me start thinking of, if you've got problems on both sides, are you going to have problems on upper and lower limbs? So I would, it's a whole area of trying to understand, is there a link with making corrective responses of the arms, and is it going to impact the legs? And then it gets into the whole thing of balance and issue, because of course you need balance control with your legs, but if you're going to fall, you better have good coordination of your arms if you're going to want to grab out and get some help. So yeah, it's a great question. I hope people work on it. That's part of the reason to get the robots out. Hopefully there's somehow they'll be of value for lots of people.
Wonderful. It's a few minutes past 1. So if there are any final questions, please type it in for now. Otherwise, we can wrap up. Thank you again for a wonderful talk and fascinating discussion afterwards. I think we have to invite you again one more time to talk about the process of commercializing your technology and taking it out of the lab into the clinics. That would also be of interest to many people.
It's a challenging thing, but yeah, it worked. We started slow and slowly built and did organic growth. I don't think we would have survived if we went deep in fast. So that'd be great. I hope I can get down. Maybe in the fall or winter when things get improved.
Sounds great. Thank you again for a wonderful talk. Greatly enjoyed it.
Thank you very much.
And everyone, please join me in a final virtual round of applause, thanking Dr. Scott.